रियल टाइम क्वांटिटेटिव पीसीआर और इसको qPCR भी कहा जाता है एंड इट इज़ कॉमनली यूज्ड टू quantify the target sequence in the reaction mixture that we can know the starting quantity of the biomolecules or nucleic acid like the DNA, cDNA or RNA molecules. Under certain circumstances, it is advantageous or desirable that we must have the idea about the starting concentration of the nucleic acid. कि अगर स्टार्टिंग कंसंट्रेशन ज़्यादा है टेम्पलेट डीएनए की या cDNA की या आरएनए की तो आफ्टर पर्टिकुलर साइकिल जो कंसंट्रेशन है वो भी ज़्यादा होगी सो यू कैन से दैट देयर मे बी relationship between the initial quantity of the nucleic acid and that accumulate after particular number of cycle in the PCR reaction. So we can use this criteria to calculate the amount of nucleic acid or DNA that has accumulated after particular number of cycles. So this approach to calculate the DNA molecule after particular number of cycles was first used by Higuchi and co-workers in early 1990s. They use a chemical which is called as ethidium bromide. If you remember, ethidium bromide, it is an intercalating agent. It can intercalate between the nucleic acid bases. And after intercalation, it can even give the fluorescence. So it is highly specific to bind with the double-stranded DNA molecule. ये जो इथिडियम ब्रोमाइड है ये डबल स्ट्रैंडेड डीएनए मॉलिक्यूल के साथ बाइंड करती है और जब डीएनए के फ्रेगमेंट सिंथेसाइज होते हैं आफ्टर ईच पीसीआर रिएक्शन तो इसकी बाइंडिंग भी साथ साथ इंक्रीज होती रहती है और आफ्टर ईच साइकिल ऑफ द पीसीआर जो इसकी जो एमिशन है वो भी रिकॉर्ड होती रहती है जितनी ज़्यादा एमिशन होगी थिडियम ब्रोमाइड की या इसकी फ्लोरेसेंस की तो उतना ही हम कह सकते हैं कि हर साइकिल के बाद डीएनए की जो कंसंट्रेशन है वो इंक्रीज कर गई है तो बाय प्लॉटिंग the fluorescence with particular number of cycles, we can calculate the DNA concentration in real time. So as soon as the DNA is replicated, we can quantify it. So because of this reason, it is also called as real-time quantitative PCR. And this technique, it is more satisfactory or suitable when we compare it with other techniques like if all the cycles have been completed or the quantification of DNA after the after the fixed number of cycles have been completed. So over that technique, this real-time quantification, it is more satisfactory. At the same time, I would like to add one point that this real-time quantitative PCR, sometimes students or some workers, they can confuse it with RT or reverse transcriptase PCR, but this is not the situation. Although RT-PCR, it can also be used in the quantification of RNA by using competitor RT-PCR, but this real-time quantitative PCR, it is only primarily used to quantify the nucleic acid. So as I told you that ethidium bromide was used initially to record the fluorescence as soon as the amplification of DNA is there. But it has certain disadvantages or limitation that it can give fluorescence for both specific or non-specific molecules. To overcome this problem, DNA probes were used. If you remember, probes, they are oligonucleotide fragments of nucleic acid that can be used to detect the target genes in the sample. So in this case, this DNA probe, it is labeled with two dyes are reported system. At the 5' end of the probe, fluorescent dye is attached, and at 3' end, quencher dye is attached. Now, what is the role of quencher at the 3' end of the probe? So, suppose if it is a probe at 5' end, fluorescent dye is there, here quencher is there, and quencher, because of the close proximity with the fluorescent dye, it can absorb its radiation so that fluorescence it is not recorded. Now, what is the mechanism uh, to generate the fluorescence as soon as the new fragment of DNA they are generated during the PCR reaction? The mechanism is that this probe will bind at least downstream of one of the primer. If primer bind here, it will bind downstream to one of the primer. When Taq polymerase will start its amplification, the fluorescent dye it is cleaved. As soon as it is cleaved, it will record a fluorescence. And with the subsequent amplification, this RNA probe it is also removed. Now quencher, now it is not in vicinity with the fluorescent dye. So fluorescent dye at the 3' end after cleaving, because of the nuclease activity of the Taq DNA polymerase, this fluorescent dye it is removed and it will record its amplification after each PCR cycle. So in this way, more fluorescence, the more the accumulated DNA concentration. So what is the basic mechanism in case of quantitative PCR? Here in this diagram, you can see that here primer at the 5' end or 3' end with the template DNA, and probe it is binding here. It is having a reporter system at 5' end and quencher at 3' end of the probe. Now, if polymerization is there, then what will happen? This reporter dye it is being removed by the nuclease activity of the system, and in the next step here it is removed. Now it is free from the quencher, it will record the fluorescence and complete polymerization is there. So after each subsequent amplification, one can say that the amount or the concentration of DNA it will increase. It is the most satisfactory technique to quantify the DNA molecule. So it can be combined with conventional thermocyclic procedure so that we can introduce the fluorescent dye to develop the RT PCR so that we can know the concentration of the DNA molecule at a particular step. So this uh, technique, that is RT-PCR, we can effectively use to know the molecule concentration even in unknown sample. And here we can generate the standard curve with known concentration of DNA, and then the concentration of the DNA molecule in unknown material, it can also be calculated.